Brilliant. Hi, I'm Jen Sloan from the University of Sheffield. I'm going to tell you about our 3D photosynthesis model in rice leaves, which we hope will help to provide some insights into growing rice in the future. If I can get my thing to work. There we go. First of all, I want to answer the question, why rice? Rice is the third most abundant grain grown globally, after wheat and maize, and arguably the most important crop for human consumption. It's a staple food, which means it provides more than 50% of the calories for more than half of the global population. And it's actually most important in the poorest areas of the world. The global population is increasing year on year, as we all know. And although grain production is also increasing, the gain in yield is currently not predicted to be enough to match the increased demand. Struggling with going on a slide, there we go. When we look to the future, we also have to consider climate change. CO2 is increasing. So in the future, crop plants will be growing in very different conditions to those that they've been bred for. We're hoping that it's possible to use modelling to look into the future in a way and find specific characteristics that will improve photosynthesis in the future. Photosynthesis models to date are typically two-dimensional, just a series of equations. However, in reality, photosynthesis is occurring within a very complicated organ, the leaf. Here I'm showing you a 3D reconstruction of part of a rice leaf from confocal images. Photosynthesis occurs mainly in the mesophyll cells, and within those cells in the plastids. Rice mesophyll cells have, as you can see here, this fairly complex lobed shape. In order for photosynthesis to occur, gases have to travel into the leaf and then make their way through air spaces, through the mesophyll cells, through the mesophyll cell walls, and to those plastids where the photosynthesis occurs. Different parts of the leaf have different amounts of resistance to that gas movement. The way the light enters the leaf and moves through the leaf is also obviously affected by the 3D structure of that leaf, with different shaped and sized cells refracting that light in a different way. We're hoping to find out if there are changes that can be made to this structure, which can make photosynthesis more effective in the future high CO2 conditions. When we're looking at this sort of structure and we want to understand it, the first thing that we really need to do is measure, and measure lots of different things. So this is our system that we're using. We're using IR64 rice and indica variety. We're looking at leaf 5, when the plant is 21 days old, is a mature leaf. From these mature leaf 5s, we take leaf sections, which gives us gross measurements of the rice leaf, like interveinal distance, leaf thickness, mesophyll thickness. The team at Nottingham University help us out with micro-CT, where you can visualize the airspace versus the cells within the leaf. So on an even greater scale there, we can look at the amount of airspace within the leaf, and from that we extract mesophyll porosity. Then we've used confocal imaging to get detailed size and shape analysis of the mesophyll cells. We've used TEM imaging to look at the bundle sheath cells, the size and shape again, and the plastid, the chloroplast content. We've also looked at the mesophyll in more detail there, looking at the proportion of plastid, looking at the cell wall thickness, and measuring SM or SMES, which is the cell wall in contact with the air. We've extracted chlorophyll from those leaves, and we've also done gas analysis. So a lot of measuring. I've just got a video here to try to show you how all of these measurements can be turned into a computational model or a structure for that photosynthesis to occur within. So first of all, we've used those broad measurements to make a kind of framework for our leaf. Then we've got the bundle sheath here, and we've matched the amount of plastid in green there, the amount that we've measured using the TEM. We've got the mesophyll cells here, we're altering the size and the shape, and we've got an average mesophyll cell now, based on all of our measurements. Then we can populate that framework with these mesophyll cells we've got from all of our other measurements in order for that leaf to match the airspace from our CT measurements. Each of those individual mesophyll cells we then adjust so that the cell wall to air contact matches our measurements. The plastid volume in green here matches our measurements, and also the cell wall thickness. So I've described all of these measurements we've done and this structure that we've created on the computer for our photosynthesis model to work within. We've actually done 2 lots of those measurements because we've got 2 treatments. So in IL-64, leaf 5, 21, 28 days old, as I said, we've grown those at ambient CO2 and elevated CO2. So we're particularly interested in high CO2 for the future, as I've said. We've got all of those measurements and then we've compared the 2. Now this graph is where we're expressing the relative change between the ambient CO2, or I've called it ACO2 here, and the elevated CO2, the ECO2 leaf. This is the relative change, one compared to the other, and these are all of the different measurements we've got that feed into our model. In this graph, it's a bit of an unusual way to look at the data perhaps, so I'll talk you through it a little bit. Draw your attention to, say, the mesophyll porosity here. This bar goes over to the left-hand side on the graph, and that means that the value of mesophyll porosity is higher, about 20% higher, than is elevated CO2. Draw your attention to another one here, the cell wall thickness. This bar goes to the right-hand side, which means that the cell walls are thicker at elevated CO2 compared to ambient CO2. And as I said, we've also done some gas exchange on these plants. So here we've got some ACI curves. So this is the internal CO2 of the leaf, plotted against the assimilation or photosynthesis. And we've artificially changed the CO2, and then we've measured the photosynthesis at the different levels. We've got our ambient grown plants here and our elevated grown plants. If you're familiar with some of the face experiments on rice in the field, you might have expected there to be more of a difference here, but we're not surprised by this. We're growing our plants in growth chambers. It's quite an artificial environment for rice, and this has been seen and reported in the literature before, that their photosynthesis is fairly similar. I'm also showing you here 5PS2 plotted against internal CO2. This gives us an idea of how the photosynthetic machinery is working in those plants. And there is a little bit of a difference here between the 2 treatments. From those gas exchange curves, we're able to extract values for these important parameters of photosynthesis, which are all part of the model. And generally, if you look at this graph again, we've done the same as before. The bars on the left-hand side, the values are higher at ambient CO2. The bars on the right-hand side are higher at elevated. Generally, the photosynthetic machinery or metabolism is like tuned up at ambient CO2. Seems to be performing better. So where have we got to? We've got two different structural models of what the leaf looks like, the geometry of the leaf. And then based on previously published models of light movement through the leaf and CO2 movement through the leaf, these are then inputted into these structures or work within these structures. And then we've got a third chunk of the model, which is a metabolism, which again works alongside these two parts and within the structure that we've measured. So using these two models that we've got, the ambient model and the elevated model, we then simulated 5 ACI curves in each model. And this line here is an average of the simulated ACI curves. And the dots are the measured values I showed you before. So our model there, the line, matches really quite nicely the measured values on our ACI curve in the ambient model. The elevated model also matches the ACI curve nicely. There's a bit of a discrepancy here with our 5PS2 data, but generally we're quite happy with this fit. It's a model, you can't expect it to be perfect. So that's great. We've got a model rice leaf and we've simulated photosynthesis curves using it. How is this helping us? We wanted to answer the question, which characteristics are most important at elevated CO2 for photosynthesis? So in order to do that, we need to pull those two models apart. We've taken the ambient CO2 model and changed just one factor at a time, factor such as mesophyll cell size and shape, or the amount of chlorophyll, and swapped that number for the elevated CO2 number. And then reran the model simulations, again, repeated 5 times under each of these new situations. And this is the data from all of those simulations. This graph here I've already shown you. This is just the different measurements and the comparison between the two treatments. Here for as a reminder, but also the colours match so that you can look across to that side if you want a reminder of what these mean. So this is the change in photosynthesis caused by changing that one factor, leaf size and shape, or bundle sheath size and plastid volume, by changing that one factor from the ambient model to the elevated value. So we've got an ambient model with just one factor change to the elevated value. And I'll just look at this pink one because it's nice and obvious. That is chlorophyll amount. So the amount of chlorophyll from the elevated model is beneficial to photosynthesis, or it causes an increase in photosynthesis. And that's relevant to the amount here. Another one here that would just show you the mesophyll cell wall thickness. So this is negative to photosynthesis. So the mesophyll cell wall thickness is thicker at elevated CO2, and that change to a thicker cell wall is a negative change. So it's bad for photosynthesis at high CO2. This black line here is if you take all of these numbers, positive and negative, and get the sum of those values. So you can see that the black line, particularly at high CO2 here, is essentially 0. So there's no change, which tells us that any positives that you can see here are being outweighed by the negatives. And I'm sure you've all noticed that the main negative here are these grey bars. And the grey bars represent photosynthetic metabolism. So the elevated CO2 photosynthetic metabolism is cancelling out all of the positive changes to the elevated CO2 structure. There we go. So I'll just go over that again. Most elevated CO2 leaf structural characteristics improve photosynthesis, particularly at high CO2. There are two elements which seem to be detrimental: the increased cell wall thickness and decreased mesophyll cell plastid volume. But most of them are positive. The elevated CO2 metabolism impairs high CO2 photosynthesis. So models are models. It doesn't mean anything if you can't test it. How can we test these model predictions? These are just predictions coming out of our model. So we really need a leaf with elevated CO2 structure and ambient CO2 metabolism. And our hypothesis then would be that it is better able to photosynthesize at high CO2 because it's got all of the positives and none of the negatives. So in order to attempt to create this test system, we've got three treatments here. We've got rice grown at elevated CO2, 1000 ppm, and rice grown at ambient CO2. And then we've got a third group of plants, the primed plants, where we grow them at elevated CO2 for 21 days, at which point leaf 5's structure is fixed. So leaf 5 has an elevated CO2 structure. We then transfer this batch of plants to ambient CO2 conditions and leave them to grow for a little bit longer in the hope that their metabolism will adjust to ambient conditions. And that's exactly what we've seen. So there's a couple of enzymes here as an example. So we've got the elevated CO2 level of FBPA's activity, that's quite low in the purple. In the yellow, we've got the ambient CO2 activity. And the green ones here have been transferred at 0 hours from elevated to ambient conditions. And you can see that after 72 hours, the primed plants have a equivalent FBPA's activity to the ambient grown plant. And the same is true with SBPA's. The green dots here, the primed ones, change their metabolism to match the ambient CO2 by 144 hours. So this is great. And then the obvious question is, how do these plants photosynthesize? Can we prove our point? The problem is this experiment has been interrupted by COVID-19. We are now allowed back in the labs. This is the first thing we're going to be doing, is testing this out. In the meanwhile, we have some promising preliminary data from our collaborators in Malaysia. So these are different rice lines. It's not IR64, and we don't have the metabolism data for these particular plants. But if we are to assume that they are the same as they act in the same way as IR64, then we could say that we have here ambient grown plants and primed plants, which have been grown for 4 weeks at elevated CO2 and then transferred into ambient conditions. So we assume that their metabolism is adjusted the same as our IR64 ones did. And then they've tested the photosynthesis, and the green ones, the primed ones, have a higher photosynthesis, more efficient photosynthesis, particularly at those high CO2 levels. So this is really promising. It's not quite the perfect experiment yet because it's not on the right rice plants. And we also don't have the elevated CO2 plants to compare. But as I showed earlier, they have a very similar level of photosynthesis to the ambient ones. So watch this space for the next experiment, really. And then I think I've said most of that, but we've created a novel 3D model of photosynthesis in a rice leaf. The output from that shows that structural characteristics beneficial to photosynthesis at high CO2 are most of the elevated characteristics, and there's a couple that aren't quite so beneficial. A rice leaf with elevated structure and ambient metabolism is predicted to be optimal for high CO2 photosynthesis. And we've shown that the Malaysian primed rice leaf with this optimal combination does perform well in preliminary investigations. So on that note, just to say thank you, really. Thank you to everyone at Sheffield. Chris did a lot of the gas analysis work and some of the biochemistry. along with Rachel. Rachel, Alice and Hannah were involved in the imaging. The team at Nottingham for the CT scanning. Yi in Shanghai has done all of that modelling and all of the computer work. And these are the people in Malaysia who did those preliminary ACI curves. And that's it. Thank you very much.
Okay, thank you very much, Jen. That's really interesting stuff. So I'm going to start with a couple of questions, if I could. So, well, first of all, an observation more. You know, there's a lot of data there, a lot of parameters. So do you really, there's a huge team of you that do that, or are you doing all those parameters? How does that work?
There's quite a big team. It's taken quite a long time. I think mostly everyone is involved in doing all of it to a certain extent. In the Sheffield team, there's a lot of imaging, there's a lot of looking at images on the computer, a lot of measuring thing.
Okay.
It's quite a long time represented in this one. We've done all of these measurements graph.
Well, yes, I can imagine there's a lot of stuff there. So I'm going to take a liberty and ask another question. So you mentioned the Malaysian field experiment towards the end. So how do you grow those plants in elevated CO2 before they're transferred to the ambient? How do those guys do that?
So out there, and these were quite big experiments as well. It's quite cool. They've come up with a method of elevating the CO2, which can actually be used in the field, and it involves bags of mushrooms. And we use those bags of mushrooms to increase the CO2 within a kind of tent. It's really quite cool.
Okay, cool. Okay, so a question from Amanda Kavanagh, and she asks, how are primed plants able to adjust FBPA's and SBPA's activity? Do they produce more enzyme over the course of 72 to 144 hours?
I don't think I know the answer to that. So we've measured their activity, the activity of the enzyme, using a spectrophotometer, but we haven't necessarily measured the amount of enzyme. So I'm not quite sure. Okay. It's not really my strong point, sorry.
Okay. All right, question from Stuart Sullivan. He asks, do you or can you include effects of climate temperature changes in the elevated CO2 model?
I think it's really a really interesting question and something that we should do. For now, we've focused on one thing, but once this model is created, we could do all sorts of things and look at lots of different questions. And temperature, obviously, is something that we should be looking at when we're looking to the future. So yes, we should.
Okay. So a question from Rakamal Kasinthan. And they ask whether you checked Rubisco enzyme levels and activity in the elevated or the ambient CO2 conditions.
I'm trying to remember. It was a little while ago that we did that part. I feel like we must have checked Rubisco, but I'm sorry, I can't remember the data off the top of my head.
Okay. Well, as I said, I'll pass the question on.
Up, yeah.
You can get back to that. So a question from Ariban Roy. So if we want to have an idea for legumes and elevated CO2, what should we do? Hang on, I'm just reading this. And which is the thing that determines the selection of the genotype? How many genotypes do we need to take? Does that make sense? I think so.
I mean, I suppose with legumes, we can extrapolate out of rice. We can say if this happens in rice, perhaps it happens in legumes, and then try to test the theory. But or you could make a whole new model of legumes, but it's tricky. But I didn't quite understand the second part of the question.
I guess that's how would you select for the genotypes that you wanted to select for under elevated CO2? So I guess that plays into the first part of your answer. Okay, so a question about basically where you go from here now. So you've developed this leaf, you put all this data in. So what are the next steps you have?
Well, once we're like confident, once we've shown that what the model predicts is true, which we are fairly confident that's going to be the case, then we need to start asking other questions really and probing further. We're hoping to develop a second version of the model, perhaps with even more measurements or different measurements, looking at other things that are important to photosynthesis within that, because we were limited in what we've measured and what we've looked at. I don't know, you can always improve a model, can't you? So
there's space for more parameters, I guess, somewhere. But, okay, so I think we'll leave it there. So thanks very much, Jen. Thank you to Mark as well. Thank you for everyone who was listening in. And as I said, so in 2 weeks' time we have one more webinar. So please go onto the page and sign up for that. But thanks, as I said, Jen, and thanks very much, Mark, and thanks everyone for listening. Thanks.